All right, it is 2 o'clock, so let's get started. Just a reminder of what we've been talking about. We've been looking at protein structure, and on Friday, we were talking about different experimental techniques to look at protein structure, so X-ray crystallography, NMR, and Cryo-EM. And then we were talking about in a folded globular protein, where can you expect to find different types of amino acids? And so generally, you have your hydrophobic amino acids in the center. On the outside, you have your charged amino acids. And polar amino acids are usually on the outside as well, but they can be inside if they're in some kind of hydrogen bond. So the next thing we're going to talk about, and I'm still on the PowerPoint from Friday, are these things called motifs or supersecondary structures. So if you remember, we talked about secondary structure being our alpha helixes and beta sheets, and we also looked at some other types of secondary structure like collagen. What a motif is something that shows up in a lot of different proteins. And they're a specific combination of alpha helixes and beta sheets. So if we just look at some of the common motifs we see in different proteins, you have what's called a beta alpha beta. Now, anytime you see an arrow like this means a beta sheet. And this squiggly, well, that wasn't a very good beta. This means a beta sheet. And this ribbon, it's always an alpha helix. So beta, alpha, beta. Again, the arrows tell you direction. So N terminus, C terminus, N terminus, C terminus. You have something called a beta hairpin, which is just beta sheets that will wrap around each other very tightly. Alpha alpha motif, two alpha helixes that are interacting together. Now, to get something like this, what is most likely happening if I'm going to redraw my alpha alpha motif out here. So you have something like this. What is most likely happening is where the two alpha helixes are coming together, you probably have hydrophobic residues. And excuse me for one. All right, sorry, I had to sneeze. So you can see in the alpha alpha motif, you have these two alpha helixes that are really just coming and interacting with each other. That's most likely due to the hydrophobic effect, where you have hydrophobic residues on these helix and water is pushing those together. You have something which is called a Greek key. And so if we look at the Greek key, we have beta sheets. So we have 1, 2, 3, 4 different sheets, and they just kind of fold over. So sheet 1 is interacting with sheet 2 and 4. 2 is interacting with 1 and 3. It's called a Greek key because I guess this is what a Greek key is supposed to look like. I don't get it, but that is what a Greek key is. And talking about proteins in general, when we look at their secondary structure, you're going to have different types of proteins. You're going to have what's called an alpha protein. And an alpha protein is a protein that's made out of nothing but alpha helices. So as shown here. Also, when we're looking at like different characteristics of proteins, this barrel that's being shown, a barrel means an alpha helix as well. And the color corresponds to where you're looking in the picture. you can have what's called a beta protein, which is just made out of beta sheets, or an alpha beta protein, which is made out of both alpha helixes and beta sheets. So those are some motifs. Those are some different types of supersecondary structures that are common in proteins. And what's cool about these secondary structures, and this word's cut down here, is that some of these structures have functions that go with them. For example, this structure that I'm showing right here is called the immunoglobulin fold. And this is shown in your antibodies. So all antibodies have this kind of beta sheet that's kind of looking like a sandwich. And that's called the immunoglobulin fold because you see those in antibodies. So different motifs have different functions. And once you start studying a lot of proteins, you'll see there's lots and lots of motifs out there. Another motif I think I brought up in passing before is called the zinc finger, which can bind to DNA. So a lot of these motifs do have different functions. So any questions about motifs or anything at all? All right, let's move on. I do have a question. I'm going to skip this because I'm not going to ask you on a test like, is this protein alpha or beta protein? For this image, really all I wanted you to do is to start to look at images of proteins and start recognizing what's an alpha helix, what's a beta sheet in this representation. So just as a reminder, and I said on the last slide, anytime you see a ribbon, they're trying to show you that's an alpha helix. Anytime you see arrows, those are beta sheets. You'll see proteins represented like this all the time, and they're just showing you the different types of secondary structure. The last thing I want to talk about on this slideshow, or this from Friday, and it's something I already talked about before, is protein domain. So let's get a little bit into what an actual protein domain is. Protein domains are within proteins, and they're usually found in larger proteins. So anything that's like over 200 residues. And by the way, when you're talking about proteins and you see the word residue, that's just a fancy way to say amino acid. So proteins that are over 200 amino acids, they can fold into different clusters, globular clusters or domains. So in the image here, we're actually coloring the domains so we can see it easier, but we have an orange domain, and then we have a blue domain, but they are part of the same protein. There is just one continuous peptide bond here. It's not two separate proteins, it's one protein with two domains. And like I say here, they're almost like independent units. They fold by themselves. They often have function that's their own. Some of them don't have functions though, but some of them do. Domain's kind of a catch-all word. And the interesting thing about a domain is that it's more important what you look like than what amino acids you have. For example, I'm showing 4 domains down here. If you look at the amino acid sequence or numbers, you can see they're wildly different between each of these species, all the way from 86 amino acids to 134 amino acids. However, all of these domains do the same thing. They all have the same function. So that really tells us that it's not so much to care about sequence. It's more, what do you look like? Because really structure defines function. What I mean by that is, if you really want to know what a protein does, you need to know what it looks like. Once you have a 3D image of a protein, then you can really start to understand how it works. And you can start thinking about how it's regulated. You can start thinking about all those types of questions. However, until you actually know what the protein looks like, you can't quite study that in depth. So it's very important when studying any protein to know what it looks like, because once you have the structure, you can start saying, what is the function of this protein? So those of you who are joining me in the live session, I just have a quick, I think I made a poll for this. Yeah, so I just have a couple of true false statements here. And I have a poll. So let's see if we are following along. So 4 different statements. I would like you to answer, are these statements true or they are false based on what we have talked about? So I'll give you a minute or two to do that. If you have questions, as always, let me know. Otherwise, I will be back with the answer shortly. So think about these, look up your notes, and I'll be back. Do about 30 more seconds on this. So yeah, just take that time, come to a decision, and put your answers in. All right, so let's go over these. So proteins with the same function, but from a different species, are likely to have similar motifs. That is true. So remember what I just said, that structure really defines function. So if you do the same thing, you mostly look the same. And if you look the same, you have the same motifs or supersecondary structures. So that should be true. Proteins with the same function from different species are likely to be more similar in sequence than in structure. That is false. Again, you can get the same structure with different amino acid sequences. So at the end of the day, it doesn't matter what your sequence is, it matters what you look like. C, an effective protein motif is likely to be observed in multiple proteins. True, right? That is kind of how we discover these motifs. We see them over and over and over again because they're really good at doing whatever their function is. And so they keep popping up in different proteins. So that is true. And D, proteins with the same motifs are likely to perform similar functions. That's kind of similar to A, but that is true. Again, motifs are structure. So if you have the same structure, you're probably going to do the same thing. Right. So any questions about these true and false questions? Right. If not, we can move on to our next PowerPoint then, because that should be it from Friday, and we'll move on to the one for today. All right. So what we're briefly going to talk about today is our last level of structure, which is quaternary structure. So I'm just going to run down the different levels of structure again. So primary is the amino acid sequence. What is your sequence? N-terminus to C-terminus, that's primary. Secondary is backbone conformation of localized regions. And we're mainly talking about our alpha helixes and beta sheets there, but those aren't the only types of secondary structure. They're just the most common. Tertiary structure, overall 3D structure of one protein. And last is quaternary, is 3D structure of multiple proteins forming a complex. So to have quaternary structure, you need multiple proteins. They need to be interacting. So if you have a protein that is made out of one peptide, and when I say the word peptide, that means one unit that is N term to C term. That's a peptide. So if you're a protein that just is one peptide, you don't have quaternary structure. If you are made out of multiple peptides, like this image here, you do have quaternary structure. So this image is made out of 4 different peptides, which is shown by the colors. And if it's the same peptide, the only way to combine is through symmetry. What I mean by that is, like, if you have a protein or a peptide, let's call it peptide A, and two A's come together to make one protein, one functional protein, they can only form in one way, and that's called cyclic. So this could be an A, and this could be an A. If there was 3 proteins, the only way they could form is in this C3 symmetry, A, A, A. And what we're looking at here, this green, what that is trying to show you is an axis of rotation. So if you had 2 A's coming together, they would have to be 180 degrees apart, like mirror images of each other, more or less. While if you had three A peptides coming together, they would be 120 degrees. If you had 5 different A peptides making one protein, they would be whatever 360 divided by 5 is, which I don't, what is that, 60? That might be wrong math off the top of my head, but everything is just separated by the same degree. And it's just these what are called symmetry operators or rotational symmetries. You can get much more complicated dihedral symmetries. What's called a D2 is that you have not only this rotation that we saw in C2, you also have a flip. And this is, I find this hard to see. The way I keep track of it is I've watched this black part that's sticking up. So like one rotation, one symmetry operator is that you rotate the protein 180 degrees this way. And then I'll do my best here to do it on a 2D plane, this 3D image. You kind of imagine you're grabbing this black piece, right? And you're kind of like pulling it up to flip it another 180 degrees. And then down again 180 degrees. And then flip back up 180 degrees. So it's all this image is trying to show you is that for quaternary structures, there are limits imposed on how we can combine peptides are the same. They have to have these symmetries. And it gets much more complicated when you're talking about things like tetrahedral symmetries or cubic symmetries or whatever this icosahedral symmetry is supposed to be. It's very hard to show these on 2D image. So just keep in mind what I really want for you to get out of this is that what is quaternary structure? So make sure you know that and know that we have to follow these symmetries. All right. So just have some questions here. So let's understand the cyclic and dihedral symmetry. So what type of rotational symmetry is possible for a protein with 4 subunits? And so the numbering system goes, the letter tells you what type of symmetry we're looking at. Remember, cyclic is just going around in a circle. Dihedral is not only are we going in a circle, but we're also flipping. So if you have 4 subunits, you could say you're in a C4 or a D2. They're not the same thing. You're either in a C4 or you're in a D2. You can't be in both at the same time. And each of these symmetries are just showing you, what's going on here. I don't have a C4 pictured, but it's possible to be in a C4. For a 6 subunit, your options, if you're just talking about cyclic and dihedral, would be a C6 or a D3. So that's how the numbering system goes, right? D, to know how many subunits are in this dihedral symmetry, you take this number and multiply it by 2. The cyclic tells you right in there how many subunits there are. And so for B, if you have 12 subunits, but they're stacked in 2 rings of 6, what is that symmetry? That would be a D6. You can see that pretty well in the D3. D4 is really hard, but in D3, you can see it's really just two rings that are stacked up on each other that's doing the flipping. You either flip 180 degrees like this, that's one symmetry, or you rotate 120 degrees. So that's how how we can combine these 6 different proteins using these symmetry operators. All right, do we have any questions about the quaternary structure or any of these symmetry operators I was talking about here? All right, if not, we can move on. All right. So the next topic is, how can we tell based on amino acid sequence where different, based on amino acid sequence, like what parts of the proteins are likely to be buried inside the protein, which ones are likely to be on the outside. Just from primary sequence, we can predict, this part of the protein will probably be buried on the inside. This part of the protein is going to be on the outside. And this is called hydropathy, or the hydrophobic scale, hydropathy scale, or hydropathy, I guess is actually how it's pronounced, not hydropathy, hydropathy. So remember we talked about the hydrophobic effect on Friday and how it's a big driver of protein folding. Water makes nonpolar amino acids go onto the inside of the protein. And we can assign numbers to amino acids based on how hydrophobic they are. It's a really cool experiment. There's different ways to do it. But you probably did something like this in organic chemistry lab. The idea is you have like a solution, half of that solution's water, half of that solution's oil, and you put an amino acid in this oil and water mixture. And then you let them mix, and you just ask yourself, okay, where are you likely to find isoleucine? Well, isoleucine is hydrophobic, so you'll find it in the oil. And it gets assigned a number based on how often it is in the oil versus the water. So this hydropathy scale is, the more positive you are, the more hydrophobic you are. And if you actually look at this on the internet, you'll find different scales have different numbers for things. But just keep in mind, the more positive number you are, the more likely you are to be buried. And so by looking at the primary sequence of a protein, we can determine based on these numbers what part of the protein is buried, what part is not buried. And we can make a table like this. So what this table is, it's a running average of hydropathy scores or hydropathy scores. So in case you don't know what a running average is, those of you who, one thing I suggest is you should follow the local government on Facebook, because every single day they'll have a COVID update, which has the 7-day running average of COVID cases. So what a running average is let's say I have, I'm going to just make up some numbers. So running average would be, I want the average of these three numbers, and the average of those three numbers is, I don't know, like 4. Then, well, my next window would be these three numbers, and the average of these three numbers would be like 5 or something. You just take like, you look at a series of data and you pick a window, you pick a length of numbers, and you just slide along those numbers, and you keep saying, okay, these numbers, that average is what? These numbers, that average is what? These numbers, that average is what? For something like positive COVID tests, that shows a much accurate and more stable number than daily tests. Because if you looked at the daily COVID test, every single week, you will see a huge drop on Sundays. And it's not because people aren't getting COVID on Sundays. It's because people aren't getting tests on Sundays, or they don't process the tests until Monday. So if you ever looked at those COVID charts, it would be like steady, 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 drop, spike, steady, 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 drop, spike. And so we have running averages to get a more accurate picture. We can do the same thing with amino acid sequences. You just do a running average of these hydropathy numbers. And if you're positive, you're more likely to be buried on the inside of the protein. And if you're negative, you're more likely to be on the outside. So these little lines are kind of showing you where on average the series of protein or this series is. So it's like this part of the protein, this part of the protein, this part of the protein, you're more likely to be on the inside. This part of the protein, this part of the protein, outside, inside, inside, outside, outside. That's what those bars are trying to show, like on average, where you think that sequence of amino acids are. And it's all due to doing this scale and taking this running average. All right, so before I move on to questions surrounding this, do people have questions what this graph is trying to show you? Right, so I really do want you to keep in mind though, positive is hydrophobic, negative is hydrophilic. Last semester when I talked about this, seems like everybody memorized it wrong. Like 80% of students said, no, negative is hydrophobic. And I'm not sure where that came from, but just keep in mind, positive is hydrophobic. So what I want you to do, and I'll give you a couple minutes to do this. I want you to draw me a plot, a hydrophobic index plot or hydropathy plot for a protein that has 7 domains that go through the lipid membrane. Keeping in mind that the lipid membrane is hydrophobic. So I want you to try and draw that. So I'll give you a couple minutes to do that. Then I'll be back with the answer. But if you have questions when doing this, please let me know. This seemed to confuse a lot of people last semester because I did ask this question on the test last semester and they were really confused. So let's try it. And if you have questions about it, let me know. Otherwise, I'll come back with the answer in a couple minutes here. All right, so hopefully you had some time to attempt to at least think about it and start drawing a plot, asking yourself, what does this mean? So I'm going to follow the template down here, draw a straight line. So this line is 0. These are positive numbers, which again means hydrophobic. These are negative, which again mean hydrophilic. All right, so protein that has 7 domains that go through the lipid membrane. So I'm going to start drawing with my N terminus, and then domain 1 is going through the membrane, and then you drop. Okay, so let me explain what's going on. So usually in proteins that go through the lipid membrane, they look like this. So here's my lipid membrane. Here's my protein. 1 domain, 2 domain, 3 domain, 4 domain, 5 domain, 6 domain, 7 domain. So domain 1, 2, 3, 4, 5, 6, 7. So, and these are lipids. So any protein that's outside the membrane is hydrophilic, right? They interact with water. So in any protein that's in the membrane has to be hydrophobic. So hydrophobic in here, hydrophilic in here. Because the hydrophobic are interacting with lipids, hydrophilic interacting with water. So that's N and C term. So if I'm doing a index, this would be my N term. And this is domain 1. And then this part down here that's going towards 0. These are going outside the membrane. But remember, this is a running average. So that I don't have to go into the negative, because most of this protein is hydrophobic. So 1, 2, 3, 4, 5, 6, 7, and then my little tail's coming out. So that is how a hydropathic index for a protein with 7 domains go through the lipid membrane might look like. So now that you've seen that plot, is there any questions about that idea? All right, there's no questions. We'll move on. Now, talking about protein folding, protein structure. I mentioned this before. The hydrophobic effect is probably the most significant thing that happens during protein folding. Other effects are important as well. Hydrogen bonds help to fine tune our structures. We talked about this when we were looking at the protein folding funnel, how these hydrogen bonds can help and make the 3D structure a little bit better. You have what are called salt bridges. And if you see the word salt bridge, all that is saying is that a positive and negative group are coming together and interacting. So that is a salt bridge. They're important for holding parts of the protein together as well. They're usually on the outside of the protein, but not always. They can be on the inside. Usually, salt bridges hold different subunits together. They're kind of like gluing different parts of the protein. So what I mean by that is that if we have a protein with a sequence like this, if I have a positive residue here and a negative residue here, I can make this protein fold over because the positive and negative want to be close to each other. So that's a salt bridge, and that can be important for structure. You also have disulfide bonds. I mentioned this before, but when cysteines come together, and the cysteine side chain of a cysteine is SH. So when you have two cysteines together, what can happen is that the side chains of the cysteines can form a covalent bond. And this is called a disulfide bond again. And this can also help to hold different parts of the protein together. Now, question here, and I'll go ahead and answer it. How can pH change our quaternary structures? Well, we just said that ion pairs and hydrogen bonds are very important. And these ion pairs, hydrogen bonds, are especially important for holding different proteins together. So if you change the pH, you can break these ion pairs or salt bridges. You can break the hydrogen bond interaction. Now there's nothing holding our protein together. And so the different subunits will just kind of float away. You no longer have any quaternary structure. You don't have a functioning protein because you broke the glue that was holding it together. All right, so any questions so far about anything that we have covered up to this point? All right. Now let's look at a famous experiment done in 1957 that deals with protein folding. And this is called the Amundsen experiment. And what Amundsen did is that he had a protein called ribonuclease A, RNase A. And he took the folded protein, the native or active form, and added high levels of urea. And what urea does is that it breaks disulfide bonds. So this urea unfolded the protein, and the urea unfolded the protein. The beta mercaptan ethanol broke the disulfide bond. So BME broke disulfide bonds, urea denatured. So he took this active protein, RNase A, unfolded it, and broke all of the disulfide bonds. Then what he did is he removed the urea and he removed the beta mercaptan ethanol. And what happened was that the protein refolded. And he saw that all of the disulfide bonds were back to what they were in the native structure. And this happening by chance is 1 out of 105, because there was apparently like 105 different combinations the disulfide bonds could form. However, every single time he did this experiment, the protein refolded correctly and all the disulfide bonds reformed. And this comes to one of the big revelations when it comes to proteins. Primary structure dictates the 3D structure of a protein. What that means is a protein folds into the shape it does based solely on its primary structure. That's all the instructions the protein needs. When it's coming out of the ribosome, based on what the primary structure is, or primary sequence, it will fold correctly. Now, in truth, Amundsen kind of got lucky with the protein he chose. as the question here says, given enough time, will all denatured proteins renature? That is, if you unfold a protein, will it always reform into the native state? And that is no. One big example is eggs. You take eggs, you add heat to them to get cooked egg. You can wait all the time you want in the world. Those eggs aren't going back to the way they were before you cooked them. So mostly when you denature a protein, there's no going back. Usually it gets tangled up with other proteins and it becomes inactive and you can't save it. However, he did happen to find a protein that does renature. But the big takeaway from this study is true for all proteins. The primary sequence of a protein is what dictates the 3D structure of that protein. All right, so any questions about the Amundsen experiment or any of the information that we went over today? Whoops, didn't mean to just make a loud bang and just drop my pen. Well, if there are no questions, that's all I have for you today. We'll pick up with the last slide on Wednesday, and then we will move on to, I believe, we're talking about protein function and doing some bit more math on Wednesday. Other than that, I'll put up a homework like always. If you have questions about anything, never hesitate to email me. Otherwise, I will see people on Wednesday. Have a good week, everybody.